Hi guys, I'm glad to be taking you through this interesting course, which I'll be learning about the gene. That unit that determines inheritance and how it is expressed. So let's get started. Okay, so what makes you different from your siblings and even your friends? It's basically the gene. It's the kind of genes that you inherited from your father or your mother. Uh, and in combination with your environment. But basically, this topic is about the gene. So, after this lesson, you should be able to describe the structure of a gene, should also be able to describe the process of genetic transcription and translation. So, what is this gene? A gene is a nucleotide sequence in a DNA molecule that acts as a functional unit for the production of a protein, a structural RNA, or a catalytic or regulatory RNA molecule. So in essence, what we're saying there is that a gene is a nucleotide sequence in a DNA molecule that can produce two things. It can produce either a protein or an RNA molecule. And it is a DNA molecule. So here I'm showing the structure or the image of a chromosome, and then breaking it down into the DNA, and then breaking it down into the components of the DNA itself, the base pairs. So a gene is a sequence of nucleotides in the DNA, a string of nucleotides, and we will see what the structure of the DNA looks like. So the human genome, which is the totality of the genetic constitution of the human, is made up of just over 3 billion nucleotides long and divided into 23 pair of chromosomes, which is 46 chromosomes. So which means that we are saying that the human has 46 chromosomes, 22 autosomes, and 2 sex chromosomes. So these nucleotides are divided between those 23 pairs of chromosomes. And then the Human Genome Project, which made this available, actually has helped a lot, because it has allowed us to be able to estimate the amount of genes that are present in the human genome. And it's estimated that we have about 25,000 genes in the human genome, broken down to about 21,000 protein coding genes and about 5,000 non-protein coding, which is the RNA, different types of RNA, mRNA, tRNA, etc. So what is this DNA structure? Because you need to understand DNA structure in order to be able to understand how the gene transcription and gene translation happens. So the DNA structure is a polynucleotide of nucleic acids, which is composed of the 5-carbon sugar, the deoxyribose, a nitrogenous base, which is either can be a purine or a pyrimidine base, and then a phosphate group. Now these two chains that run parallel to each other are bonded together by hydrogen bonds. So you have two hydrogen bonds between adenine and thymine nitrogenous bases, and then you have two hydrogen bonds between cytosine and guanine nitrogenous bases. And so this structure allows the chemical information that is present in this DNA to be able to be transcribed into the required information that we want, the required product, which is the protein or the RNA. And so that's the basis of transcription. So from this image, you would see that this is a double helix, two chains running as parallel to each other, and then made up of the deoxyribose, made up of the phosphate group and the nitrogenous bases. And you can see the pairings between those nitrogenous bases and the hydrogen bonds in between. So basically, we have something in molecular biology that we call the central dogma. So it's just saying that, look, information is transmitted from the DNA into mRNA and into protein. So the process whereby the information in DNA is copied into mRNA, messenger RNA, is transcription, and the process by which that mRNA is copied into a protein is what you call translation. So the structure of this DNA and the sequence of nucleotide that it contains actually specifies the amino acid sequence of the polypeptide chains of proteins. this is basically what genetics taught. They look at how this sequence that they find in genes, how are these things expressed into proteins, and at that level they look at what happens at doing transcription and what happens doing okay, so the RNA, in this case the messenger RNA, is synthesized from this DNA template, as you can see in this this central dogma image on the right here, where the DNA is being transcribed into an mRNA, which is the messenger RNA, which is now translated into a protein. So the sequence of the mRNA is decoded or translated into a protein molecule. Now this process actually happens in different locations. You have transcription, which happens in the nucleus, and then you now have an export of this messenger RNA into the cytoplasm, where translation actually happens. So translation actually happens in the ribosomes, which are the sites for protein synthesis, and all these processes involves some types of RNA, which is you have the messenger RNA, you have the ribosomal RNA, you have transfer RNA, you have small nuclear RNA, which are involved in RNA splicing, but we'll get to that. So what is the gene structure? What is this structure of genes? Basically, this is the structure of a typical eukaryotic genes, and what you find here is that you have a 5' region, which is there, you have a 3' region, which is there, and then you have the transcription initiation, you have the translation start codon, you have the translation stop codon, you have introns, and you have exons. So basically, in eukaryotes, the genes are interrupted by one or more introns. These introns are the non-coding portions of a gene. These are the ones that they don't actually form any sequence. These are the regions that actually do not code for part of the protein. But then you now have the exons, which are the actual coding portions of gene. They are the actual part that you would find in the messenger RNA, which will be translated into a protein. And so basically in the human genome, few genes actually have no introns. In fact, almost all genes have introns, and in fact you haven't have several genes, a gene having several of these introns, making very larger percentage of the gene, and we will see an example. And so you have the largest human gene, which is the dystrophin gene, which is about 2,500 kilobase pairs in length, having greater than 95% of intron material, and the actual part of the gene, which is exon, which is the coding portions, is actually just about 0.5% material. And so what happens is that because these regions are non-coding, they have to be cut out during the process of protein synthesis. So before this mRNA is being moved out into the cytoplasm, those introns have to be cut out in a process that you call splicing. So but then before that, if we looked at that structure of the gene, you find that you had a region that you call the promoter region, and you have some other regions that are there. But then these regions, you have what you call the promoter region, which actually specifies where transcription will begin. It is the site that actually specifies where transcription will begin. And so it allows the RNA polymerase, which is the enzyme that is actually involved in the synthesis of this messenger RNA, to be able to function, to be able to move along the gene and carry out this process of transcription. And so you have other sequences on the gene. For example, you have enhancers, silencers, you have locus control regions. These are all involved in gene regulation. And so basically, because earlier it was always thought that these introns, these non-coding portions of a gene, are actually junk DNA. They don't have a function. But now we know that many of these genes are actually involved in regulation of genes. So basically, here we have the whole process of protein synthesis, the process of transcription, which happens in the nucleus, and then you have the process of translation, which happens in the cytoplasm. So basically what happens is that you have the unwinding of the DNA exposed to the RNA polymerase. So here you have, this is the RNA This is the RNA polymerase that is synthesizing the messenger RNA, and then it synthesizes the mRNA, giving us the sequence of the mRNA, synthesizing the nucleotides that is joining together. Now, after this synthesis of this mRNA, once it reaches the stop codon of the gene, it stops transcription, and then it dissociates. Now, but what happens is that this mRNA, at this point, is called a primary mRNA transcript, or a primary RNA transcript, that must be processed before it becomes a functional mRNA. So what happens is that RNA processing involves what? It involves three things. It involves 5' capping actually. 5' capping, which is the addition of a modified guanosine nucleotide, and then it also involves 3' polyadenylation, the addition of a poly-A tail, like you find here, poly-A tail. It's about addition of about 200 adenine nucleotides, and then you now have splicing. So you have three things that happen during mRNA processing. So after this processing, you have what you call the functional mRNA product, which you now call the mRNA, which now is being exported into the cytoplasm to the ribosomes. This is the site of protein synthesis. So basically, the ribosome has two subunits. It has two subunits: a smaller subunit and a larger subunit, and it has three sites: the E site, the P site, and then the A site. So what happens is that this mRNA actually binds to this ribosome. It's like, as you find here, it binds in the center of these two subunits, and then fills the three sites that you have there: the E, the P, and then the A site. So what happens is that this sequence of nucleotides that you have on these ribosomes are being used to bring the correct amino acid sequence. So what happens is that the information there is being read 3 nucleotides at a time, a codon, and so this codon specifies particular amino acids. And so when you have a specific amino acid, so here what you can see here is that you have UGG. Now you have a transfer RNA. Remember I mentioned transfer RNA before. You have a transfer RNA that brings an amino acid that is complementary to this sequence, to this codon here. So this is a transfer RNA, and then it brings an amino acid that is attached here. So what happens is that, let's say it's reading the first 3. So you have the first amino acid here. This is being expunged off. You have the next one, that is say for example phenylalanine. It carries the phenylalanine amino acid, and then it moves along like that. So as one gets expunged, the thing moves and shifts to the left. That's 3 nucleotides at a time. So this chain, for example, this growing polypeptide chain that you have here, will then be transferred to the next amino, to the next transfer RNA that is carrying another amino acid. So, and that is why you would find that this chain will be continuing to be elongated until it reaches the stop codon, the stop signal on the messenger RNA, and then this synthesis of polypeptide stops, and then the going polypeptide can then become a protein. It folds up to become the protein, and so basically that's the process of transcription, which happens in the nucleus, and then translation, which happens in the cytoplasm. So the sequence of this polypeptide, this going polypeptide that we've just formed at the ribosome inside the cytoplasm, actually forms a structure, and this structure is specified by the sequence of the amino acid that you find in there. So you have different kinds of amino acid that have different properties. Some are polar, some are non-polar, some are aromatic, some are hydrophilic and all that. So based on these properties and based on how they are arranged along this polypeptide chain, that determines the structure. So you have the primary structure, which is just the primary sequence of the amino acid in the polypeptide chain. You have a secondary structure, you have a tertiary structure, and then you have a quaternary structure, which is, for example, the structure of hemoglobin, which is the pigment that is involved in oxygen transport, is a quaternary structure. Or the you have beta pleated sheet, which is an example of a secondary or tertiary structure, alpha helix, beta pleated sheet, and all that. And so sometimes also these proteins need to be modified for them to be able to be active or to be useful. So there may be the addition of carbohydrate parts or other parts, other molecule parts, cofactors, and all that. So you have, for example, glycoproteins and all. So these proteins sometimes need to be modified, and so they are modified, they are processed at the rough endoplasmic reticulum and then exported through the Golgi complex. Now this gene, as you have it in there, that is producing protein, have to be regulated. So you have different control points of gene expression. You can control gene expression at the process of transcription. You can control gene expression at the process of mRNA processing. You can also control gene expression at the process of mRNA transport outside of the nucleus. You can control gene expression at the process of translation. You can even control gene expression at the process of protein, of the post-translational modification of the protein molecule. So, but the most efficient, or the most one that does not waste cell energy, is transcriptional processing, because you regulate the gene in such a way that it's not even transcribing at the beginning. Instead of having to transcribe the gene and having to now be having your control when it has already transcribed, it has already produced mRNA, having to degrade some mRNAs and all that. That's will require a lot of cell energy, and so it's not like they're all useful, but transcriptional processing, transcriptional control seems to be better. So in summary, what we've discussed is that the cell must use the information contained in this DNA to make proteins. Now, these proteins are highly essential biomolecules involved in life processes. Now, the use of information in this DNA to make proteins is divided into transcription and translation. And so I said that the transcription occurs in the nucleus while the cytoplasm occurs in the cytoplasm. Now, these proteins that have been synthesized may need to undergo modification, and so what you call the post-translational processing of protein molecule. Now, the structure of a protein is based on the amino acid sequence of the protein. Now, the expression of a gene is regulated at different control points. Now, this is what we'll discuss during the next class. And now I believe that you would have understood the concepts of gene expression, the concept of transcription, and the concept of translation. Just the vocabularies that you've encountered in this lecture and lesson. You have the genes, the DNA, the exons, the introns, which are the non-coding portions of a gene, and then you have splicing, the removal or causing out of introns of a gene, and then you have RNA polymerase, not DNA polymerase, RNA polymerase, enzymes that are involved in the process of transcription. So I believe that we would meet in the next class. So bye bye class, and have a nice day.